All right, welcome back. Oh man, look at them. Look all fresh and clean shaven compared to my last videos. Um, so we're gonna get back into this cell interaction stuff that we kind of been talking about in the last few videos with compaction and the cells, the conditional specification and things like that. So we're going to talk a little bit about the interactions between these cells as they're dividing, and so that brings us to cell adhesion. So in our little ChatGPT AI image here, we have some cells here that we have all these. It looks like kind of a cell wall, and this stuff kind of sticking it together. I don't know what's going on, but we'll talk about coherence and some of these cellular molecules that interact between the cells as they're growing. So briefly, there's signaling factors in addition to the adhesion properties, right? So knowing your neighbor, right? So for conditional specification, knowing who your neighbor is and getting that information to determine, oh, I need to be muscle because all my neighbors are muscle, or neurons because all my neighbors are neurons. That information can come in two different ways, right? And so we have, well, three different ways, but we'll talk about two of them here. So juxtacrine signaling, so that is additional, essentially knowing your neighbor physically, right? So this is local signaling, where we have these membrane bound protein receptors that bind to extracellular proteins or the membrane bound receptors of another cell. So they can either bind, usually there's a transmembrane, right? So they have transmembrane domains, so they go inside to outside of the cell. So it's a interior and exterior protein, so to speak. So you can see this in these this diagram here. Here we have two different types of binding. We have homophilic binding, where one protein will bind the exact same protein in another cell, or we have heterophilic, excuse me, where one protein will bind a protein of a different type. And then we also have this protein down here that's binding like an extracellular matrix or something that's not a membrane-bound protein. We also have paracrine signaling. So this is signaling where the cell excretes signaling proteins, or ligands in this case, or ligands. And then on another cell, there is a receptor that is a transmembrane protein that will bind that ligand and cause usually a phosphorylation or some sort of chemical cascade that ends up changing gene expression, etc. So those can go much further distance. They're not super long distance. The third type of signaling that is usually not quite in early development, but is important for later development, is hormones, right? So hormones are a little different than ligands in that they go a much greater distance, right? And so that's kind of through the circulatory system, testosterone or estrogen or anything like that, progesterone. When we talk about sexual development, which we'll talk about later, those are much longer distance than, or can travel longer distance than paracrine factors. Okay. So getting back to how our talk on conditional specification. So not only does your neighbor influence who you are, but you also tend to group with them once you've been determined to be the same type of cell, right? So in this experiment, there's these amphibian cells from the epidermis and from a neural plate, right? So they took these, excised them. I believe this is from a frog. I don't have that written down. I believe it's from a frog. But what they did is they took these two different tissues that were developing, so they're already determined. So this is a neural plate cell, and this is epidermal cells, or presumptive epidermal cells. And they put them in they changed the pH of the solution that they're in. And so it caused all these cells, all those cell adhesion proteins to break their bonds. And then they're all free floating. So you've got a big mixture of these. So then they changed the pH back. That allowed them to, those chemical linkages of these Juxtracrine factors to kind of reestablish, and allowed them to coalesce back together, and the tissue spontaneously, you can kind of see it in my picture here where you have it in your slides, it reorganized back into the original stuff. So instead of having like this, originally it kind of looks like, oh, you know, you just have a mashup. Well, just spontaneously, if you disorganize stuff and then take whatever stimuli that was away that caused the disorganization and put it back, let them go back together on their own, they'll organize back into their own tissue types, which is kind of cool. So this same thing held true for all sorts of different combinations. So they did it with epidermis and mesoderm, mesoderm and endothelium, epidermis, mesoderm and endoderm, etc. And every time they mixed them up, they always would organize back into their relative tissue types as soon as they took away that stimuli that was causing them to mix up, like changing the pH. So this is called a selective affinity. And that's that certain cell types have either a positive or negative attraction to other cell types. So for instance, the inner surface of the ectoderm has a positive affinity for the mesoderm cells and a negative affinity for the endoderm cells. So this was ectoderm, mesoderm, and endoderm. Ectoderm, mesoderm, and endoderm. So let's look at this picture and let me read that again. So the inner surface of the ectoderm, endoderm. Oh no, ectoderm. Maybe this isn't depicted here. Well, we'll use this as an example. So the mesoderm has a positive affinity for both the epidermis and for the endoderm. The endoderm has a positive or an affinity for the mesoderm, but a negative affinity for the epidermis, and the epidermis has an affinity for the mesoderm and a negative affinity for the endoderm. So these two don't want to touch each other, yellow and blue, right? Red wants to touch both of them, that layer. So these different affinities are what causes this organization to happen. Is some tissues are positive, or some cells that are determining or becoming tissue have positive affinity for certain tissue types and negative for another, and that causes these layers to occur, right? And so this is from the we call this the differential adhesion hypothesis. So this actually it's kind of a hypothetical syllogism. So it's without getting too into the weeds about it, you don't have to like completely understand this. I took a logic class and this stuff is really interesting to me. But it's a if A then B, if B then C, therefore if A then C. So for example, if it rains today, I will wear my rain jacket. If I wear my rain jacket, I will keep dry. Therefore, if it rains, I'll keep dry, right? And so you can kind of use these type of technique or this theory for these tissue layering, right? So if A is attracted to B and B is attracted to C, then A would be attracted to C, right, etc. So the core concept of this differential adhesion hypothesis is that different cell types have different adhesive properties or varying levels of stickiness is a good way to put it. The differences in adhesion drive cells to self-organize and sort themselves. So we saw that when they take them apart and they'll go back together in their respective groups. Cells arrange themselves to maximize the strength of adhesive bond and minimize free energy in the system. So they're going to do whatever, it's like thermodynamically favorable, right? So if you've taken physics and stuff, you might know a little bit about thermodynamics or chemistry, where you're going to do whatever is the most favorable energy state. And so it's very similar to a very similar process to how immiscible liquids, so like putting oil and water, separate based on surface tension, right? So if you put oil and water and they're different charges, right? One's polar, one's nonpolar. Then you get these, bubbles of oil in the water. Because thermodynamically that's the best state, and eventually all the oil will aggregate together in the water, whilst there'll be a layer on top of the water, right? Well, depending on the heft, I guess, of the liquid. But anyway, so that's a very similar concept here with these cells, is kind of like they want to self organize in that same way. So in the molecular basis is that there's these cell surface proteins called cadherins, and we'll talk a little bit about them here shortly. So here is this cadherin. You can see that these are membrane spanning proteins. So there's these adhesion recognition sites on the end here, and a calcium binding site, and a phosphorylation site at the on the inside here. So that helps with the signaling purposes, right? So you have a phosphorylation site that can help to send signals across, and then it's this binding to actin filaments through these catenin proteins. Let's see. What did I miss? Anything? So if you remove the cadherin or block its synthesis, this causes the cells to disaggregate to become single cells, like they'll stop clumping together. And because it's linked to this actin cytoskeleton, like I said, this allows signal transduction too. So this relays to the nucleus who your neighbors are type stuff. Okay, so the amount of cadherin helps to determine the morphogenesis, right? So there's a linear relationship, as we can see here, between the strength of the bond and the amount of the cadherin. So the higher surface tension is related to the higher number of cadherins per cell. So if there's thousands, like this is thousands, 200,000 cadherins, you're gonna have a very sticky cell. If you have only 20,000 cadherins, then you're gonna have a much less stickier cell. So cells with a lot of cadherins will be attracted to other cells with a lot. So here the green cells have have lots and grouped together, while the red cells are left to the outside. So down here, so green has a ton of cadherin, so they're all gonna kind of exclude the red because they don't have as much, and so the red just kind of are on the outside looking in, literally, and the greens all group together because they have stronger bonds, because they have more charge with those cadherins and more stickiness, right? So the cell sorting can occur with different cadherin types. So if the red cells have more than the green, let's give me a pointer here. If red has more than green, then red will sort together and green will be on the outside. If red and green are equal, we kind of get a mismatch, right? Because there's no difference between the positive and or like one's not more sticky than the other. So they all are fine sharing it. And then if green has more stickiness than red, green will be on the inside because they stick together a lot tougher than the red. Okay, so not only is the quantity important, so the number of cadherins that we talked about, but so is the quality or the type and the timing of the expression of the cadherins. So there are different types. For example, N cadherins, which is a type, as we can see, we have E and N are two different types that we have. They appear in the mesenchymal cells just before the start of cartilage formation, and then disappear right after. So if N cadherin is blocked by antibodies or something, or changes in pH, or the gene doesn't express or something like that, then cartilage won't form. So they're there to kind of set up where the cartilage is supposed to be, and then the cartilage forms and then they go away because their job's done. But if they never existed, then cartilage doesn't form because they don't have that information, right? So the type can matter as well. So R and B cadherins do not mix well, and if they form two piles of cells, so that's the example up here where we have R and B cadherin. So they just don't like each other. So they kind of stick together, but they're very cliquey, right? Like in high school, you have two groups that did like each other. And then, so the boundaries that these form are necessary for proper morphological events. So, for instance, all embryonic cells initially express E-cadherin at the beginning here. And then those cells will eventually become the neural tube, and they'll lose the E-cadherin expression and start expressing N-cadherin instead. And this separates those cells from non-neural tube cells. So initially they all express it, then when they need to separate and become a different tissue, they express N-cadherin, and it kind of allows them to form that barrier. So if you block the N-cadherin synthesis, there is no proper border formation between the skin and the nervous system. So here you can kind of see we have this nice formed, we'll get into what the neural tube is coming up during gastrulation and organogenesis, but it's a tissue layer here, and if you block N-cadherin, it just becomes like there's no distinct layers. It's just kind of one big jumbled mess. You can kind of see where it initially was expressed. The E-cadherin was starting the formation of this neural tube, which you would see over here, but it just becomes a big mess of gobbledygook. So that is cell adhesion. So it's important to know when we talk about the conditional specification, what the actual actors are. And these are these cadherin molecules and catenins, this transmembrane proteins that allow signaling across, and the amount of cadherin or the type kind of determines stickiness, and that helps these layers build. So trying to keep it 17 minutes on the dot. I hope you enjoyed this one, and we'll keep on in the next one.